Hi.
Hi, how are you?
I added something called an edge measurement. Edge point is a probe. It's an edge probe to try to find the height of my structure, and it's better, but it's not like it's somewhere there's the math that goes with this. You know how it adds, and it says integration 4. I have no idea what that is. So I just wondered, what is integration 4? And then you put it on addition and it does the same thing. So I don't know.
Yeah. I don't know.
And I just got turned down for funding to go on a course about COMSOL because it was $700 US.
Yes.
for 3 or 4 days. So they just, they said, oh, no, not a required course.
Yeah.
So it's not funded, so.
Yeah.
So there's no help to a help desk there, so.
Yeah.
Unless you want to pay $1,000 Canadian just for the privilege of talking to someone about it. I don't know.
Right. Yeah.
They deserve to have themselves priced out of the market completely.
Oh, yeah, yeah. I think they've been taking full advantage of their monopoly power as being like the standard multi-physics platform. So.
Yeah, so they maybe should just.
MATLAB's kind of like that too, but yeah, not quite as shameless as COMSOL.
Yeah, well, I've just, anyway, I did find the study, the one that's MATLAB oriented programming, so I would like to do that next.
Okay.
Yeah. Anyway, so can you see me?
Well, your camera's off. Yeah.
Is that what it is?
Yeah, I mean, it just went off. It was on before. I don't know.
Yeah, no, I may sign off and come back in.
Okay, all right.
Because.
Yeah. All right.
Yeah. Okay, if I can see my screen, I'll sign off.
Okay.
Anyway. All right, we'll see you in a bit.
All right, see you. Hello, Pakhi.
Hi, hello.
Hi. All right. So do you have updates?
Yeah, so over the last week I was working on pre-processing the data that I would be passing on to the graph in the MLP, like I had mentioned in the last meeting. So maybe I can share my screen.
Yeah.
Yeah, is it visible?
Yes.
Yeah, so if you remember, I like in the last meeting I told I was trying to run some code that they had put in their repository. The NDP people, they had made a new comment, but like I was facing some errors, so I just dropped that. I just wanted to see what new implementation they had done. They had used some reinforcement learning algorithm in the new commit. So like I dropped that for time being. So like I was just generating data. So I'm planning on like using time series data from like the XLSX file that you had shared with me about the birth and death timings. So using that, I'm like trying to generate the data as of now, and like these nodes. Basically, like I'm removing the parent because like it gets divided into two, and then adding both the children instead. So like at each timestamp, what the graph sort of would look like. So I'm currently working on like the data preparation. It should be done in a day or two, and then like from tomorrow, I am expecting to start like building the model as well.
Okay, that's good.
Yeah.
So this is just data processing, and then you would plug this into the neural developmental program.
Yeah.
Okay.
Like those functions also I worked on like editing some modifying some functions to like meet our needs, like what we need to do with like the growing neural network. They are using they are trying to mirror like the XOR gates and a few models they're using, so I like edited those functions as well over the last week. So
Okay.
yeah.
Yeah.
Once I have like the data ready, I'll have like more sense of what I need to add to those functions, the NDP functions. So yeah.
Okay. So
Yep.
last time you said you were having trouble implementing the program that they have for NDP. So they have a software there.
Yeah. The in the NDP, like in their repository, they had like recently made a new commit in which they were using like reinforcement learning. So I found that interesting. So I just wanted to explore what it is. But like when I tried running the code, I was like running into some errors. So like I dropped that.
Okay, so you did get them resolved, or do you have a workaround?
No, like I mentioned, right? I like then got to like starting my work as well. I'm looking into it at the side. It requires me to install like some libraries and also that was taking up GPU in collab, and like I don't have GPU in my laptop, so I like preserve the GPU for making our model for now. Yeah. Yeah, I'll get back to it and like let you know if it runs.
Okay, yeah. Well, it's going to be the next step. So you've got some time on that to get that all sorted out. Yeah, we've often run into problems with GPU time and GPU access. So this is a common problem, and we don't really have a good answer to it. But I don't know. I don't know what to tell you there. But I guess it's probably something you can do just kind of with the collab resources. But yeah, let's kind of think in terms of like GPU scarcity and how to overcome that, I guess. Okay.
Yeah.
Okay. Well, thanks for the update, Pakhi. Looks good. Hello, Dick. How are you? Hi. And we talked to Susan about her console misadventures or adventures or whatever. Right. So the first thing is that sometimes a lot of the stuff we do in DevoWorm and OpenWorm gets cited. So we write a paper and then people cite that paper. And if you use something like ResearchGate, it alerts you to when your paper has been cited. So Dick sent me this email alert he got about this OpenWorm paper. This is a paper that was published in 2018. It kind of goes over OpenWorm in terms of the different components. And it's been cited probably about 150 times by now. So it's, a decent paper in terms of citations. One of the recent citations, and, the citations are really diverse. Like you see all sorts of different citations for it. Not a lot in the traditional C. elegans literature, but actually a lot in the sort of the AI space and modeling space. So that's interesting. But this is the latest citation of this paper. This is Discovering Neural Policies to Drive Behavior by Integrating Deep Reinforcement Learning Agents with Biological Networks. So this is in Nature Machine Intelligence and with these co-authors. So I figured, I'd go get the paper and look it over and see kind of what they're citing and, what they're doing. So this is the paper here, Nature Machine Intelligence, Discovering Neural Policies to Drive Behavior by Integrating Deep Reinforcement Learning Agents with Biological Neural Networks. So they're using this deep reinforcement learning paradigm, which is, of interest, for a number of problems. I think it has a good relevance to biological problems because organisms to learn things will often engage in some form of reinforcement learning. And so reinforcement learning, to recap, is something that kind of came out of behavioral psychology. You think of B.F. Skinner's work and some of the other behaviorists of the early to mid 20th century. That's kind of where it comes from. And then Rich Sutton, who actually has a degree in behavioral psychology, brought it into computer science and brought it into AI applications. So we're doing reinforcement learning. You're basically training an agent on some environmental stimuli, and they have these policies that get basically chosen. You have to choose a policy to address what's in the environment. So when you're learning a policy, and you're learning from a set of policies which one is the best. And so you reinforce the learning with different types of things. You can reinforce it with sort of a conditional stimulus. You can reinforce it with some sort of reward. So there are a lot of ways to reinforce the information that's being learned, and then that's included in the policy. So the policy gives information about, like, what you're doing, how you respond to a certain stimulus, what the reward is, what the conditional stimulus is, and so on. So it's a really, this is something that's been very hot, especially in the last couple of years, with respect to, kind of solving a whole new class of problem. So, and then there are these different variants. So in my other group, we're working on a project where we're looking at multi-agent reinforcement learning. So this is like reinforcement learning across a group of agents or a population of agents. Deep reinforcement learning, of course, combines sort of deep learning with reinforcement learning, and this whole idea. So that's what they're doing here. So there's different variants of reinforcement learning, and the way they approach is a little bit different, but it's useful, I think, to get getting some really interesting results. And so this paper, they talk about deep reinforcement learning and how it's been successful in a variety of domains. But it's not yet been directly used to learn biological tasks by interacting with a living nervous system. So this is something where now people are taking these advanced reinforcement learning techniques and trying to map them back to biology and to behavior so that we can learn something about those domains. So it's really interesting, the circuitous route that reinforcement learning has been taking. We're, kind of taking it as a computer science tool and then now reapplying it to organismal learning. So as proof of principle, we show how to create such a hybrid system trained on a target finding task. So this is, again, a hybrid system, which they'll lay out in the paper. And they have this, target finding task where the organism has to find a target, and there's some, criterion for success. And then there's a policy that describes how to make that decision. And then that policy is reinforced through some sort of, which one is sort of the best at doing it, and it's rewarded, and then it's chosen, and so forth. Using optogenetics, we interface the nervous system of the nematodes, the elegans, with a deep reinforcement learning agent. So they've taken the nervous system, which is basically, I guess, simulating the connectome, and then adding a deep reinforcement learning agent to that. Agents adapted to strikingly different sites of neural integration and learn site-specific activations to guide animals towards a target, including in cases where the agents interfaced with sets of neurons with previously uncharacterized responses to optogenetic modulation. So they're using this technique, it's this optogenetic technique. So if you're not familiar with that, it's where you have some sort of, you have cells where you have some sort of protein, and you use light spectrum to activate the neurons. So you activate this fluorescent marker, you activate the protein, it's basically turns on and off the neuron. So you can actually stimulate specific neurons or populations of neurons with an optical signal. And, this is something that's very well controlled when you have a system where you can specify the protein that's being turned on or off, and you just pick the right sort of protein for that, and you can get some you can actually modulate the nervous system in that way. So this is they've done a lot of work with optogenetics in terms of characterizing the responses of different neurons to this stimulation. So in C. elegans, of course, we know what every neuron is and what it does, and people have worked on this problem for a while. So they kind of know what the responses are to these kind of stimuli. And, the stimuli aren't necessarily binary. So it's not that you just turn it on or off. You actually stimulate the neuron, and you can get this modulated response. So it's an interesting approach as well, sort of incorporating optogenetics with reinforcement learning agent, computation, and then of course doing this in a connectome where you know all the cells. Agents were analyzed by plotting their learned policies to understand how different sets of neurons were used to guide movement. So basically, each agent learned a policy, and that policy basically told them how to guide their movement, what kinds of neurons to activate and so forth. And then we want to understand how different sets of neurons were used to guide movement. So some organisms, when they made a successful movement towards the target, certain neurons were activated, and that corresponds to a policy in the reinforcement learning space. Further, the animal and the agent generalized to new environments using the same learned policies in food search tasks, showing that the system achieved cooperative computation rather than the agent acting as a controller for a soft robot, which I guess this is where they just talk about how this system generalized to new environments. So basically, you can use the same learned policies in food search tasks to different types of food search tasks, and there's this generalization that's available to it. So you can use sort of this kind of approach to control a soft robot. You can use this kind of approach to show the sort of generalizability. Our system demonstrates that deep reinforcement learning is a viable tool, both for learning how neural circuits can produce goal-directed behavior and for improving biologically relevant behavior in a flexible way. The first thing I want to do is find the actual citation of the OpenWorm paper. Okay, it's number 50 actually, so that's... the first thing I wanted to show was that we have this citation of OpenWorm. So this is a section here actually that's really interesting. So this section kind of focuses on what deep reinforcement learning agents are able to do, and then what C. elegans nervous systems can do. So deep reinforcement learning tends to require large amounts of data. For instance, agents learning to play Atari can require thousands of hours of gameplay to achieve good performance. So this is from citations 33 and 34, and there's a whole literature on sort of using Atari games as a benchmark for reinforcement learning algorithms. So that's probably what they're citing. So they're basically saying that, to do this sort of deep reinforcement learning effectively, you need large amounts of data. And just kind of mentioning what kinds of, if you're using a benchmark, how long it takes to train the model. It was infeasible to collect thousands of hours of recordings in our environment, and unlike video games or physical systems with reliable dynamics, adequate computer simulations of the C. elegans nervous system and its behaviors are not available to generate training data. So that's 50. So yeah, and with OpenWorm, you don't really have, it's not really, when we have computer simulations, it's not really the same thing as allowing us to do this. So I don't know if that's like a positive thing or a negative thing, but in any case, I think the point is that, even when you're simulating a nervous system like C. elegans, it isn't necessarily enough to sort of, successfully train a deep reinforcement learning agent and have it perform all in a benchmark. So, they needed to facilitate algorithm development and reduce the amount of data needed to learn the target finding task. So they needed to do some actual training in actual worms in a wet lab. And then following approaches in supervised learning, the data were then augmented during training by randomly translating and rotating the animal in a virtual arena, approximately the size of a 4-centimeter diameter evaluation arena. So that's what they're doing. They're training the worms to behave, and then they're getting this training data. The deep reinforcement learning agents were unstable and prone to sudden performance drops, similar to previous work. So they have previous work on this. In simulated environments, such performance crashes can be monitored using evaluation episodes in the exact environment used for testing. In our environment, evaluation episodes were impractical because they would have required many more times the amount of data than were used to train agents. So this is interesting. They're training these deep reinforcement learning agents. They're doing this kind of from, these observations, these direct observations they're making, which, often happens when you're doing modeling of like an experiment. That if you want very specific types of data, you often have to get those data through a very specific type of experiment. So a lot of the data we've talked about is very general, and it allows you to look at like very general questions. But for something like this, you probably need a very specific data set. And then what they're pointing out here is that in training, these deep reinforcement learning agents were unstable and prone to sudden performance drops. So this is in the supplementary figure 1. It's not really clear why that would be. It's interesting because, so they used a number of regularization methods to help with stability and found that ensembles of agents were effective for our environment. So they used ensembles of agents. So there are a lot of methods that you can use for this. So I'm going to go through some of the figures and see what they have here. So this is the basic setup of the experiment. You have this interface between the biological neural network and the artificial neural network. You have this target finding task. So here's a nematode doing a target finding task, and you're mapping between these. You're using the data to train this network. You have the C. elegans and its surroundings and this reinforcement learning agent. So there's this observation reward that happens when the C. elegans does this target finding task. That information is fed to the RL agent, which then issues an action. And then we have this agent, which is it's an actor-critic model where you have this observation of an actor and a critic, and there's feedback from the critic to the actor that helps the actor output an adaptive action. That's where you get your different policies from and the policies that are selected. Then there's this optogenetics aspect where they actually stimulate the neurons based on this information from the RL agent. So they do the simulation of the RL agent. They then use optogenetics to influence the C. elegans nervous system. And they use this LED camera. So this is where their arena is in this plate. So we're dealing with very small scales. And, we have the stimulation here delivered by the LED. And it's delivering a signal to the plate. And so that's basically what they're doing. And then there's a lot of stuff. So they're using this ensemble approach where they're kind of, simulating multiple agents and sending a single signal to the C. elegans. So that's basically what they're doing in the experiment. This actually gives a little bit more information about it. You have these different targets. This is their task. They start in the middle. They go towards targets in different directions. If basically you stimulate the C. elegans with a random light pulse. So what you can do is you can do this untrained paradigm where you stimulate the C. elegans nervous system with a random pulse, a random optogenetic pulse. And the worm just kind of goes around from their starting position. using, I don't know if it's Brownian motion, but it's definitely this unformed trajectory that doesn't really do anything. It doesn't move towards the target is really the point. So random light stimulation, you can shut down or excite certain neurons, and it kind of disrupts the nervous system, the motor system. With no agent, meaning that you just have like this pulse on the neurons, I guess you think are going to be involved, or there's no stimuli whatsoever. The worm kind of explores its environment, maybe moves a little bit closer to the target, but doesn't really do, anything significant. And then with the agent, they show this trajectory that kind of wanders around and meanders around, but it makes it to the target. This is with the agent and then the proper stimulation. And so this is F as the known policy. So this is with the known policy in the reinforcement learning paradigm, where they go from their starting position up to the target. They don't quite get to the target. And so this kind of describes the different ways that they kind of thought this problem out. So they're using this reinforcement learning agent. They have these known policies to get from the starting position to the target. And you notice there's a lot of noise in the movement, but it basically makes it towards the target. And so we have these, performance metrics here. This is closest distance to target in centimeters. This is, with agent is here. Known policy is here. So these are two different conditions that result in some sort of success. And you see that the distance to target is minimized. And then you have these others, no agent and random light. And those are both sort of higher in terms of how close they make it to the target. So definitely having the reinforcement learning policy in place has an effect. Reinforcement learning has an effect. So that's in figure 2. Then in figure 3, the system could successfully navigate different optogenetic lines to a target. So these are, of course, with agent, random light. So random light, they kind of go around they actually move away from the target. With agent, they kind of move away from the target but come back towards the target. No agent, they kind of move around, maybe towards the target, but don't make it to the target. It's just kind of like a biased random exploration. So this is line 4. As line 5 is with the agent and random light. So there's some role here, I think. So this is for different lines of C. elegans, I believe, where, they're just testing that line to see if they're, how they perform. And so I guess, you could say with agent, they make it to the target, they explore around the target. Random light, they maybe, they don't really do anything significant. And then no agent, they kind of make it towards the target, but they don't make it to the target. And then line 6, you see similar results. And then the system learned to navigate different optogenetic lines to a target with neuron-specific strategies. So this is where you have specific neurons, as we know in the C. elegans nervous system. We know that there's specific neurons that play a role in the motor circuit, and so we can stimulate those. We can use policies to stimulate them in combination. But basically, we have these different ensembles here, and they give you specific movements. So this is the body angle and the relative head angle. Just shows you kind of their change for each. I guess this is like all possible movements here, and then they show this count of. They show the relative head angle for different lines and the line. So they show the body angle and the relative head angle at plus or minus 180 degrees. So as the body changes relative to the head angle, what does that look like? And they have these kind of visualizations in phase space so that show the wild type here where it's kind of blurry. It means it's kind of at random. Line 1 behaves a certain way. Line 2 and 3 behave a different way. Lines 4, 5, and 6 have this somewhat similar to line 1, but it's a little bit less structured. So you get these different movement behaviors, basically, that you can extract from the data and plot like this. So they look at the mean L2 difference between the individual agents in the ensemble and the final trained ensemble. And then D is where all agent policies for line 1 through 6, which is up here, and an agent trained on wild-type data where there was no possible successful policy. So they show this, the wild-type data had no successful policy, and the rest of these were basically just all agent policies combined. And then finally, this agent policies can predict agent performance on other lines. So this kind of shows this agent training line where you have these 3 agents. They show the different neurons where they're turning on specific neurons. So this one you have AIY, line 2 you have AWC on, and line 3 is here, and then you have it goes into this for sort of phase space, and you can see that based on different neurons and different strategies that you have these different patterns. So for AIY, for example, you have this kind of striped, these, I don't know, they're almost like mock bands where, diagonally, where they have this pattern, and then agent 2 and 3 have a similar phase space, and that's based on this AWC. And, with reference to this, we have this movement circuit. It's rather relatively small. It involves neurons like AIY and AWC, and they're connected in certain ways. So when you activate or suppress one neuron, it has an effect on the rest of the network, but those effects are different. And that's kind of what you see here. This portrait is, that's why it's variable like that. And it affects performance. So this is just basically showing, again, some of the data where they have these different lines. So you have a line 1 animal with a line 2 agent. So what you're doing is you're mapping the results from the animal to the agent. The agent has this phase space that represents the output, and then you're cross-combining different strategies. So basically, you're taking a line 1 animal with the line 1 intervention with agent 2 and what that phase space looks like. So you're combining these 2 together. And you can see you can make combinations of all 3 of these. And that's what they do here. So this is where you have a line 1 animal with a line 2 agent. This is the target. This is the starting point here. And then it explores actually away from the starting point. The same thing with line 1 animal with line 3 agent. It explores away from the target. Line 2 animal with line 1 agent. It explores away from the target, but not as much. Line 2 animal with line 3 agent, it actually goes towards the target. Line 3 animal with line 1 agent, it goes away from the target again. And then the line 3 animal with line 2 agent goes around, sometimes away from the target, but ultimately reaching the target with significant exploration. So this means that some of these policies will match, or in the agents will match the intervention with the the different neuronal interventions they're making here, and sometimes they don't. So it's an interesting study, and I can't say that I really understand exactly how these map to the different policies, and because this is at this point it's kind of like they're combining so many things. It's kind of hard to see what the mechanism is here, but in any case, they're getting some interesting results. So yeah, that's good. And so I think that's all I'm going to talk about for that paper. Hello, Morgan. I see you made it. So this is an article that I don't remember where I came about this, but this is an interesting article about optimal transport. And someone saying everyone should learn optimal transport, part 1. This is Mufan Lee or Bill Lee. And he wrote this blog post on optimal transport, which is something that, it's an optimization problem. So it's interesting from a machine learning perspective, from a computational perspective. But it's also something we have in biophysics. It's actually a very broad topic. So there's all sorts of transport in cells and in neurons and everything else where things are being transported within the cell, in between cells, etc. And so this is something that, he kind of sells the idea of optimal transport. And so it's kind of walking through what this is and why it's important. And I just thought it was interesting, so I wanted to bring it up in the meeting. So he says, in my opinion, optimal transport is a seriously underrated topic. I think part of the reason is the way optimal transport is often introduced. And it's introduced usually as an optimization problem or a metric on probability distributions. So it's like, we think about optimal transport, we think about the optimization aspect, that it's this optimal process that's, optimized over time. There's this, we think about this in terms of being a metric on probability distributions. And there are a lot of things we can do with that in biophysics. So, this is actually of interest to probability distributions and some of the things that we're interested with there. In a series of 2 posts, I'm going to present a message that is well understood by experts, but often missed by the uninitiated. And that is optimal transport gives us calculus on the space of probability distributions. So this is important for a lot of theoretical machine learning, as well as a lot of biophysics that we want to do. We want to look at the calculus in the space of probability distributions, as well as this sort of optimal process. While I will begin by discussing on a high level what I meant by this message, I will later present 2 concrete examples using optimal transport as a tool of calculus that will make this very clear. These 2 examples will form the basis of the 2 blog posts. So he gets into some of these things. The first one is from general but weak to specific but powerful. And so he actually asks the question we should review while we want to have a calculus in the first place. The space of probability measures naturally forms a set, which in the language of set theory is very general. So if you have a probability measure, you have this sort of set of things at any given time that are present. However, when studying probability measures, we often desire more structure. For example, we often want to equip the space with a topology of weak convergence. And then this is the math for that. In simpler terms, this defines what it means for a subset of P, which is this probability measure, to be open, and the structure of open sets define a topology. So, and so most of you reading this post have probably seen optimal transport defined by a variant of the following problem. And so, we basically have 2 probability distributions and a set of all couplings, which are joint distributions of the marginals listed here. Then we can define the 2 Wasserstein distance, so we use a distance metric here. We can indeed show that this distance metric is a metric on the probability space, the space of distributions with second moments, and this provides a specific notion of distance. So we're building from this set of this sort of probability metric or probability space. We have these sets that define what's there at any given time. And then we're moving towards a topology. And then we're moving towards distance metrics. So we have these sets. We have the sets around a topology. There's a distance between the sets on that topology. And so we have distance. Consequently, this forms a metric space, as we have discussed. Now, as many of you know or suspect, the metric structure is much more powerful than a topology, and there are many more theorems we can prove about this metric spaces than topological spaces. So topological spaces are generally just kind of like spaces with some sort of form to them. It might be a curvature, it might be a flat surface, it might be something like a graph topology, but we need to have a metric space to make sense of what that is. Otherwise, we're just kind of pointing in a certain direction and saying, there's another set over there or to the left. It's very disorienting. So we needed a metric space to describe, sort of the location of things relative to one another. At the same time, Euclidean spaces like RD are also metric spaces, but they're far easier to work with compared to metric spaces. key gap between Euclidean and metric spaces is differentiability. So this is an important thing for like machine learning and deep learning, is differentiability. So you have this distance metric, you have this metric space. On the metric space, then you have sort of a distance between two things, and then you want to be able to differentiate over that distance. So you can know sort of the different moments of, say, like, we saw those trajectories in the first paper. So we might want to say, take that trajectory, knowing that we have a starting point and a target, knowing that there's a distance between them, knowing that that's on a metric space, and then saying things about, say, not only the position, but the acceleration and the velocity and other properties of that trajectory. So this is why we want to have differentiability. But we also want to be able to say maybe about things in time. So we want to know, like, over a certain amount of time, how does this trajectory unfold? Because if we just show you a trajectory without any temporal information, it doesn't really make a lot of sense. It's just a squiggly line. But if we can differentiate over time, we can know that over this time, this period of time, the agent traveled this amount of distance, and with more time, the agent traveled more distance. Those distances are often not uniform, so sometimes the agent speeds up and slows down over that trajectory. And so this is why we want differentiability. In fact, spaces with a metric and a differentiable structure have a name. They're called Riemannian manifolds. And particularly, we need a special inner product called the Riemannian metric, or G, and an affine connection, which can be thought of as identifying a unique second derivative. The above discussion can be summarized in the table below. So this is our notation for space and then for a structure. So we have, for example, we have sets, which are this notation P. There's no structure to a set inherently. So the set is just a collection of things. Then there's this topological space, which is P and tau. And that's an open set. So the open set allows for things to move around, for it to divide and move around in that way. It's on this topological space, so the set isn't just like a single collection of things, but there's this sort of notion of spatial distance or of temporal distance, but it's a space that doesn't have a metric. So then we have metric spaces. The metric spaces are P and D, and the structure there is distance. So now we have a distance between those open sets. So when we have one set that maybe becomes two sets and drifts apart, we can then detect what the distance is between those two sets, and we can say if it's positive or negative, or if it's to the left or to the right. And then finally, the Riemannian manifold, which is beyond a metric space. These three notations here is triplet, and that gives us calculus. So once we have distance on a metric space, then we can do calculus. We can do differentiation. We can do other types of integration. We can do other types of operations that are, I guess you could say, calculus-like, if not true calculus. So basically, we can do, this leads us to this mathematical representation. Now, to fully develop the theory of the Riemannian manifold structure of optimal transport, sometimes called autocalculus, requires a whole book, which I won't do here. However, I will sketch out a concrete example next to illustrate an application of the calculus tools that optimal transport provides. And so it kind of talks about in optimization, for example, we care about the convergence of gradient descent. And of course, we care about gradient descent in machine learning and in deep learning as well, because this is the way we kind of figure out what our, how to minimize our loss in the algorithm. So as the algorithm explores the problem space, it's going towards the minimal point in that gradient. And so there's this gradient descent process. But also in optimization, we also have a gradient descent process. We have these gradient flows in continuous time. So we need these trajectories, we need this metric space, we need these Riemannian manifolds. It turns out the exact characterization of the exponential convergence is due to this factor here, cited in this citation. We will define the inequality as follows. So there's this inequality, the PO inequality, Polyak and Lozachowicz, And so this is where it was cited here. Once again, the importance of this PL inequality is that it exactly characterizes the exponential convergence rate of gradient flow. And so then, suppose xt is the solution of the gradient flow ODE with respect to the potential of this and the initial condition of this. So here we have the equation here. Then F satisfies a PL inequality with constant alpha larger than 0, and if and only if gradient flow converges exponentially fast for all initial conditions. While this may seem a bit mysterious at the moment, this characterization is that derivation is a straightforward exercise in calculus. In fact, we can quickly sketch the forward direction. So we can sketch out the time derivative. We can look at that with respect to the PL inequality, and then we can look at this in terms of Gronwall's inequality, which is the desired exponential convergence result. So you can do, this is pretty much mathematics and kind of finding equivalence and equivalencies, but this is why this is interesting. So that's one point. The second point is on the convergence of Langevin diffusion. So a widely studied process in physics, statistics, and machine learning is the Langevin diffusion, defined by the following stochastic differential equation, which is here. So these are differential equations that have a stochastic component to them, where BT is a standard Brownian motion. So for the stochastic component here, you have a Brownian motion that we saw in the last paper, where there's this movement around a central point, and it's stochastic in nature, so it kind of squiggles around that point. And so, we plug in Brownian motion to this stochastic differential equation, and this describes Langevin diffusion, and then effectively it's a gradient flow plus Brownian noise. Now, there are many reasons why Langevin is nice, but one particularly helpful property is the stationary distribution is the Gibbs distribution, which is here. And this leads us, of course, to thermodynamics and Gibbs free energy. It's rare to be able to characterize a stationary distribution of a stochastic process in such an explicit and nice closed form. We can use this to show many desirable properties. However, when the time t is finite, how good is Langevin at approximating the stationary distribution? So they move from sort of this diffusion process to stochastic differential equations to a Gibbs distribution that describes these probability distributions. And so this gives us this exponential convergence. And then we can look at exponential decay and entropy. So we can start to move towards entropy and move towards Kullback-Leibler divergence. So KL divergence, of course, is used widely in machine learning, but also describes sort of the divergence of two systems under entropy. And so, there are these connections between physics and thermodynamics and machine learning and generalized learning. So there are a lot of interesting connections here. And I'm not going to go on too much more about this, but I think, kind of give some final words here. I think the most insight I gained through learning optimal transport is the fact that calculus is powerful. The drastic simplification of several well-known PDEs, or partial differential equations, using Wasserstein gradient flow, which we didn't talk about up here, but it follows from the discussion we had about Wasserstein distance, and so maps it to this gradient flow, is an incredible breakthrough. We have just observed one important application of it for the Fokker-Planck equation. I hope this post is already sufficient motivation for everyone to learn optimal transport. So that being said, he has another blog post that is on the topic, and I won't go through that blog post. I just wanted to go through the first one. In the next post, I intend to explore the geometric aspect of the Wasserstein manifold interpretation, and then discuss an application of mean field neural networks. So this is where you get into neural networks finally, and sort of the idea that there are these physical, they have these physical properties to them, and they can be used as physical mechanisms for learning. Furthermore, I intentionally left out many important steps and details. In particular, the Wasserstein gradient can be computed explicitly. There are many nice references to optimal transport today that cover these details, but I strongly recommend VIL03 as an accessible introduction. So this is a sort of a computer science type of way of putting references in a paper. It's basically the first three letters of the authors. So BGL14 is Pakhi, Gento, and Ledoux, 2014, so it's BGL14. This is VIL03, which is, if we go down here, it's a Villani, Topics in Optimal Transportation, the American Mathematical Society. So we have other, we have Polyak gradient methods for minimizing functionals. We have Otto, the geometry of dissipative evolution equations, the porous medium equation, a topological property of real analytical subsets, the variational formulation of the Fokker-Planck equation, that's in the SIAM journal, Jordan, Kinderlayer, and Otto, and then this paper by Pakhi, Gentile, and Ledoux, Analysis and Geometry of Markov Diffusion Operators. So there's a lot of heavy math in that, but I think it's like kind of describes this sort of the practical usefulness of optimal transport and how it leads us from single like sets of objects or collections of objects to calculus and to looking at things like gradients and optimization and beyond. So I know that was pretty heavy stuff. I didn't expect to get very deeply into it. So there's probably a lot there that I didn't really hit on. Do we have any comments or questions about any of that? Yeah, it was, yeah. But that's, like if you go into theoretical machine learning, they really get heavy with math, especially also with biophysics. But I think, there are some takeaway messages there where you go through this sort of process of, looking at a problem and, even like thinking about like the biophysics of development. We have like these collections of things that we're observing, like cells or proteins or whatever. And then you can build upon that. You can say, okay, we're modeling this collection of things. And then there's this biological process. It could be like cell differentiation. It could be protein diffusion. There are a lot of things that you can model using these methods. And then, you build this sort of metric space. You build these other tools. And then you can build models of like diffusion, models of gradient descent. And you can get, you can optimize your model. And then you can end up with some pretty sophisticated analysis of that. So, it's really an exercise in model building as well. The stuff I showed several weeks back on building an optimal cell, that's the kind of thing they're doing there, except a lot of it's under the hood of software. So a lot of the mathematics are in the software, and you plug in data and you plug in parameters and you run it, and then you get these answers. And the same is true with multiphysics as well, where you get these you don't have to deal with solving equations, you just have to kind of know what they do and then run them and then get an answer. And so there are all sorts of techniques then you would use besides that, and modeling, like coarse graining or optimizing the parameters or whatever. But, all right, well, thanks for attending. And Susan.
Okay, no, I didn't have anything else. And
okay.
Thank you for that overview. That looks interesting.
No problem. Okay, thank you. Bye. Have a good week.
Bye.
Thank you. Bye. I think I lost. What am I? All right, so I'm going to draw out on the board a little bit about what we've learned from this optimal transport article. So basically what we're trying to do is we're trying to build probability distributions which give us some sort of structure or structural information. All right, so that's what they're getting at. And so structure just simply means if I have like a set of data that exists in clusters, right, I can divide this up in a number of ways. I can look at different clusters. I can look at the probability that some point is in a cluster. So I can define a cluster in some way using a centroid. And I can define whether, I'm classifying some point in a structure. But I can also say, have these sets and say, what kinds of, how likely is it that any one of our particles are within that set, and we can have statistics about the distribution of the set. So you can say a lot about structure, spatial structure, and even temporal structure, because these sets evolve, and we'll see this in a minute. But basically what we want to do is we want to find structure, say in transport or in some other system. So if we think about like a pipe where we have transport going through the pipe, we might have particles along for the ride, and they might be going down this pipe at a certain rate, but the distribution of particles is not even. It's sort of clustered like this in some ways. You have some particles that are closer together than others. There isn't this sort of uniform distribution of particles, and so that can lead to things like turbulent flows, because you can have barriers within the pipe. You can have eddies form, and you can go from this sort of laminar flow that I show here to more of a turbulent flow. And so this is where the structure becomes amplified. So that's what we're trying to look at with respect to this. So, he kind of gives a sort of a process of thinking about different mathematical tools and how that relates to structure. Thinking about different mathematical tools and how that relates to, sort of getting at that structure. So he starts with the notion of a set. And of course, a set is a collection of things. That collection of things are, pretty generic, but it doesn't have any structure, at least inherently. A set can have structure if it's, as I showed you, like if it's part of a cluster, or if it's maybe something that has, some relevance relative to the rest of the system. But in general, when we define a set, it's a collection of things, and it doesn't have any structure. Then he talks about topological spaces, which of course they're sort of a collection of open sets. And open sets don't have structure inherently, but they do allow you to have, you can think of sets as being like these static things. All right. And open sets are maybe more dynamic. So if we think of our favorite example of cell division, we can think of like the mother cell as being maybe like the initial set, and then it's an open set in the sense that whatever's in that original set can bifurcate into daughter sets or daughter cells. And along with those cells, of course, you have all sorts of things like proteins and cytoplasm and other things that go along and sort of, change their identity in this fashion. So once you have this topological space, which results from having these sets that are sort of relational, and you have the space, you have to have some sort of topology that they operate on. Then we can move to metric spaces. So metric spaces of course, are going to have a little bit more relevance to analyzing structure. So we have our distance metric, which takes these open sets and allows us to make statements about how far apart they are, or how far they've kind of diverged. So you can see this where you have these different sort of calculations of distance. And so the distance metric is, allows us to say things relative to one another. So, you can have topological spaces that have these relational aspects, but the metric space actually allows us to put numbers on a distance instead of just saying that it's diverged or that's to the left or that's to the right. Then you have these Riemannian manifolds. And these are parts of a metric space that allow us to sort of derive a calculus. And so once we have these distances between these open sets on a metric space, we can then take, say, this trajectory and analyze it by differentiating or by integrating and so forth. So we can do all sorts of things that you can do with calculus on these trajectories. And so this is where it becomes sort of important for reconstructing our probability distribution, because then we can say different things at different properties. It could be a Riemannian manifold that allows us to get information about like divergence. It could be about sort of the temporal structure. So, for example, like I said previously, the divergence of these two open sets are not uniform. One might diverge at a faster rate than the other. There might be leaps and starts and different things. And I'll talk about a paper in a little bit that kind of goes over the reality of what we have in cell biology with respect to how cells and cell populations behave as soft active materials. And then finally, we have our Gibbs distribution, which allows us to do two things. It allows us to reconstruct a probability distribution. So we have a probability distribution. How are things distributed in time or in space? It's usually a spatial array that we're interested in reconstructing. And then we have our thermodynamic aspect. And so this allows us to sort of figure out how things are distributed in terms of energy. So, we usually have in StatMech, we'll have this energy landscape with minima, and we have particles that kind of start at an initial point, and then they kind of minimize their energy accordingly. They move to the lowest parts of this energy landscape. And then we calculate the distribution of all those points. There's an underlying distribution here, and that tells us something about sort of the energetics. But there's also a probability distribution, which is connected to this. But in something like machine learning or deep learning, the probability distribution isn't explicitly connected to the energetics. So we can use the Gibbs distribution, or we can use this kind of model to calculate a probability distribution, to calculate gradient descent, without worrying too much about the energetic aspect. We just assume that things are optimized, things that, they're minimizing their loss or minimizing their energy appropriately. So we don't worry about the thermodynamics explicitly here, but what we do have is we have a model of a probability distribution and a model of thermodynamics all in one place. And of course, then we can do the calculus, we can do, talk about properties of structure, and then do the analysis for that. Now I'm going to highlight a very long biophysical review article, just kind of going over the table of contents and talking about its relevance to cell biology, and then the relevance to what we're talking about with optimal transport. So optimal transport, of course, has relevance to cell biology, both in terms of because there are transport processes that we're interested in. It's also, useful in modeling because then we can take biological processes and abstract them to mathematical structures and mathematical tractability. But then we also have this aspect of biology as a soft active material, or biological properties as soft active materials, and that's what we'll talk about in this article. So this article is called Life Off the Beaten Track in Biomechanics, Imperfect Elasticity, Cytoskeletal Glassiness, and Epithelial Unjamming. So this is where they talk about elasticity, the elastic properties of tissues, and the biomechanics, that you have a lot of hysteresis. So you don't have like perfect elasticity, but you do have these properties that are interesting in terms of, if you deform tissues, how do they respond? Then you have the cytoskeletal glassiness. So this is where we're talking about spin glass models and the properties of glasses and linking that to how cytoskeletal components behave. And then you have epithelial unjamming. Of course, we've talked about jamming phase transitions in the past and how those jamming phase transitions mediate migration of cells in sheets and in populations of cells that are migrating in development. So as I said, this is a Biophysics Review article, and the abstract reads, textbook descriptions of elasticity, viscosity, and viscoelasticity fail to account for certain mechanical behaviors that typify soft living matter. So we have our typical types of behaviors in sort of inorganic systems. But those mechanical behaviors aren't really, don't really hold up when we're talking about tissues. And I think Susan's experiencing this of trying to model biotensegrity as opposed to merely tensegrity, where you have a mechanical system, like a structure, like a building, or some sort of mechanical structure that exhibits tensegrity. When you try to apply it to the biological context, it doesn't really work as well. That doesn't mean that that model is wrong. It just means that we don't have the right parameters. We don't have the right concept of how these things work in soft living matter. Here we consider 3 examples. First, strong empirical evidence suggests that within lung parenchymal tissues, the frictional stresses expressed at the microscale are fundamentally not of viscous origin. Second, the cytoskeleton, or CSK, of the airway smooth muscle cell, as well as that of all eukaryotic cells, is more solid-like than fluid-like, yet the elastic modulus is softer than the softest of soft rubbers by a factor of 10 to the 4th to 10 to the 5th. So this is where, we kind of talk about these systems. We have these sort of properties that are not exactly consistent with what we think of when we kind of model them. So lung parenchymal tissues, they're frictional stresses that are expressed at the microscale. These are not of viscous origin, so we have, we have to model this appropriately. We also have the cytoskeleton of the airway smooth muscle cell, and as well as all eukaryotic cells, that are more solid-like than fluid-like. So they've undergone this sort of jamming phase transition, and they have the solid-like aspect. Yet the elastic modulus is very soft, and so you would think you would have to model it as a fluid, and in fact you would model it as a solid that has a very soft elastic modulus. So this is something that, again, if you use something like a multi-physics platform, you may or may not be able to calibrate your models appropriately. Moreover, the eukaryotic CSK expresses power law rheology, innate malleability, and fluidization when sheared. So it has all these properties, these power law responses that have a lot of sort of rare events or rare modes in them. This sort of malleability and fluidization when sheared that don't occur in comparable sort of inorganic, physical, or mechanical contexts. For these reasons, taken together, the CSK of the living eukaryotic cell is reminiscent of the class of materials called soft glasses, thus likening it to inert materials such as clays, paste slurries, emulsions, and foams. So this is a type of glass called soft glasses. We often think of like clays or emulsions or foams as being comparable. So again, we have these models. We know how they behave, but this is closer to what we're looking for than our typical sort of mechanical models that we use. Third, the cellular collective comprising a confluent epithelial layer can become solid-like and jammed, fluid-like and unjammed, or something in between. So like I said, we have these jamming phase transitions that when you get enough particles in at a certain density, it jams the material. So if you have a loose collection of particles, it behaves like a fluid, it flows. When you get enough particles sort of at a critical mass, they become jammed and they behave like a solid. And a lot of systems, like with cell migration or with material properties, they can go in between these two modes, so they can become jammed and unjammed at different phases of their function and their development. So we have these aspects of the epithelial layer. We have and sometimes they can be solid-like and jammed, or they can be fluid-like and unjammed, or they can have this intermediate phase of behavior, which is, I guess, the unstable phase of the phase transition. Esoteric though each may seem, these discoveries are consequential insofar as they impact our understanding of bronchospasm and wound healing. So, wound healing is another component or another example of where you have a lot of these kind of behaviors other than development. So wound healing, you have a lot of migration of cells to the injury site. You have a lot of different types of cells operating there. And you have a tissue physics. You have to integrate that into an existing tissue, often with some sort of topological aspect to it. So if you have a wound on your skin, in an area where your finger is curved, or you have a fold in the skin, then that's something that those cells have to respond to. There's some interesting connections there between something like wound healing, the soft material aspects of it, and then what we were talking about in the optimal transport article, where they talked about you can go from, say, like a topological space to these metric spaces to these models that allow for calculus and probability distributions based on thermodynamics. So it's an interesting kind of extension of this. Cancer cell invasion and embryonic development are also sort of characterized by these soft active materials. And so, yeah. Moreover, there are reasons to suspect that certain of these phenomena first arose in the early protist as a result of evolutionary pressures exerted by the primordial microenvironment. So these phenomena that we see in soft living matter, or tissues of soft living matter, or cells of soft living matter, they go back very far in evolutionary history. So they probably occurred in early protists. And so what that means is that very early in the evolution of life, we get cells. The cells can be, these rigid structures. They can be, they could be mechanical, I suppose, but they're not. They have these soft, active material properties. And so it's really interesting to think about how some of these things evolved. So how some of these types of materials evolved and why they behave in the modes that they behave in. And so the primordial microenvironment sort of forced a lot of these things to be selected for. And so this is an interesting aspect of this. We have hypothesized further that each then became passed down virtually unchanged to the present day as a conserved core process. So these are properties that are conserved widely in the tree of life, and for good reason, because it allows for these biological systems to be flexible and to do things like self-repair and other types of aspects as well. These topics are addressed here not only because they are interesting, but also because they track the journey of one laboratory along a path less traveled by. So I guess that means that they've engaged in a lot of cutting-edge research when this wasn't as mainstream as it is now. So they have a lot of really interesting things in the table of contents. They talk about the imperfect elasticity of lung parenchymal tissues, and this has relevance to tissue friction, which is where the tissue moves against itself, or in different parts of the tissue move against different layers of tissue. There are classical descriptions, of course, which are more or less inadequate, and so you need to be able to make change those models. And then there's this concept of tissue hystericity, which is, of course, hysteresis in tissues and its dynamic response. There's hysteresistivity, smooth muscle contraction, and actomyosin cycling. So hysteresistivity means that it's resisting hysteresis. This is in the context of smooth muscle contraction and actomyosin cycling. So we have this model, the HHM computational model, that talks about this, and they introduced that in chapter 3. Chapter 4 is perturbed equilibrium of myosin binding. 5 is a hard day in the life of a soft cell. This is where they get into the idea of soft cells, where, we talk about what the hardness or softness of the cytoskeleton is, because the cytoskeleton is, composed of a number of composite macromolecules that kind of get configured in a certain way. So, when we're talking about biotensegrity, we're not really talking about the softness or hardness of the cytoskeleton. We're talking about the structural stability of the cytoskeleton. So this is something where we can explore the cytoskeleton in terms of, maybe it doesn't behave like an inorganic mechanical system. Maybe it has its own sort of properties. And so, there's also this aspect of mapping the cytoskeleton to soft glass models that we have in physics. And of course, we have this plastic response, of course. So it's not just this static cytoskeleton. We have a cytoskeleton that's plastic and it changes with respect to forces. So unlike our inorganic mechanical systems, it's capable of self-repair. It's capable of this sort of deformation that it can recover from, and so forth. And then, of course, we have the role of water, which we don't think of too much. But of course, it's extremely important in biological systems because biological systems, especially tissues, are immersed in water. And it's not something we worry about that much, at least not in terms of, not something we worry about too much in inorganic mechanics. Usually water is maybe compartmentalized in those kind of models, or it has a specific interface. And so this is something that we have to think about. So stretch fluidizes the cytoskeleton. That's chapter 6. Chapter 7 is mapping physical forces that drive collective cellular migration. So then they talk about cellular migration, epithelial jamming, and unjamming. And so there's this phase transition that exists. And this kind of talks about how this sort of was discovered and sort of the dynamics of it. There's a theory, unjamming transitions, cell shape in the vertex model. And so they talk about this model in the fruit fly embryo, the genomics of the unjamming transition model, and the unjamming transition in cancer progression. So they give a nice set of model organisms and model systems here. Then they talk about eukaryotic evolution, which I think is probably the most interesting chapter, at least to me, soft matter physics and eukaryotic evolution. So they talk about the cytoskeleton as a soft glass, and this is, of course, the basis for sort of thinking about maybe biotensegrity as being fundamentally different from inorganic mechanical tensegrity. You have rigid or soft, which means just kind of like what's, if you look at the origins of life, should organisms be rigid or should they be soft? Should they be, mechanical or should they be sort of their own thing, biologically soft, as we think about it today? So, we think about like molecular machines. We think about cells as perhaps enclosing those molecular machines. And what's to say that that shouldn't have been maybe like a very rigid, hard shell? Or should it have been an adaptive soft shell? Those are all things that happen very early in evolution, at least in eukaryotes. There are advantages of being soft, of course. There are soft ecosystems and strong selective pressures for the softness. And then these conserved core processes that lead to this sort of cytoskeletal softness throughout the tree of life, or at least through eukaryotes. So it's interesting to think about what those conserved core processes are. There are, a lot of molecular pathways that are involved in making these soft materials that, we have yet to learn. Soft materials are very important, for example, in inorganic chemistry and in biophysics, or in other parts of physics. There's a soft matter physics field, which is distinct from biophysics. And so, you could learn perhaps how to design materials if you understood those core processes and sort of how proteins are built and so forth. Why is biology innately messy? And so they talk about some of these aspects of the messiness of biology. So some of that is that it's poised on the edge of chaos, that there's always in this state of like kind of the transition between jamming and unjamming. There are a lot of other sort of things at the edge of chaos that are interesting in biology. We'll get into that here, and there is a lot of literature on this actually, but that's one of their reasons. And then, of course, this need to be plastic, to be malleable, and to be able to remodel on demand. So this is where we get into this aspect of plasticity, but also being at the edge of chaos. Kind of an interesting foray into this sort of considering why things are the way they are. So they kind of, to finish up, we'll talk about this table here. And this is sort of their own reflections on their own work. But they talk about these eight consequential discoveries in basic biophysical sciences, which I think are really interesting. And so, let's go through them. So the first one is frictional stresses expressed within lung parenchymal tissues are tied directly to the tissue elastic stress and not to the overwhelmingly fashionable, but nonetheless mistaken, notion of a tissue viscous stress. So they're thinking about this in a different way. They're thinking about tissue stresses on lung parenchymal tissues, friction within the lung parenchymal tissues, so how things move against one another. And so people typically use this measure of tissue viscous stress, which is fine if it's a viscous material. It describes certain aspects, but you can actually look at the tissue elastic stress, and what they're arguing is that's a better measure for frictional stress. Number two is that frictional stress expressed within activated airway smooth muscle, which is ASM, precisely tracks and thus comprises a window into actomyosin cycling rate, hydrolysis of adenosine triphosphate or ATP by actomyosin cycling, and unloaded shortening velocity. Basically, frictional stress within airway smooth muscle actually allows you to look at some of these aspects of metabolism. Number three, activated ASM in the healthy lung attains a length that is equilibrated not by a balance of static forces, but rather becomes lengthened dynamically in response to load fluctuations associated with the act of ordinary spontaneous breathing. So, in your typical static model, you would balance out forces and you would observe them and you'd say, okay, there's an equilibrium. What they're saying is that there's actually more of this dynamic equilibrium that is dynamic over time. So if you have load fluctuations in time, you have this dynamic response to those load fluctuations. And so rather than using a static model, you would use a dynamic model. And so that's where this differs. So you can actually get a better account of what's going on using a dynamic model. So, sometimes things operate in a sort of a static equilibrium. So for example, when you're breathing normally, that The balance of static forces model describes that pretty well. But during a spontaneous asthmatic attack, ASM can become excessively shortened, stiffened, and frozen into a solid-like static muscle state called latch. So this looks at things where, outside of the normal mode of operation. So if you have a spontaneous asthmatic attack, the tissue actually undergoes this jamming phase transition, where it changes its nature from a fluid to a solid, or a solid-like state. And so this is an interesting point when you're looking at these out of equilibrium instances. Number 4, the cytoskeleton, or CSK, of the living cell expresses certain universal features, including small stiffness, power law rheology, and innate malleability, which we talked about, reminiscent of soft glasses. So these are the properties of the cytoskeleton that they've observed, very distinct from other types of materials, and certainly from inorganic mechanical systems. Number 5, in response to transient stretch and hold, the cytoskeleton of the living cell expresses prompt nonlinear strain stiffening, followed by slow stress relaxation. So you have this nonlinear response to stiffening and relaxation. So this is the cytoskeleton's response. And this is very different than something like transient stretch and hold. But in response to transient stretch and unstretch, with zero residual strain, the cytoskeleton reveals a simple universal response, namely fluidization. So if we think of the cytoskeleton as sort of a solid network, we can use transient stretch and hold to, sort of probe what it does. And what we get are these not only these nonlinear modes of response, but we get this fluidization. So it behaves sort of differently under this assay. And so this is an interesting sort of aspect of cytoskeleton. Cytoskeleton fluidization is attributable in part to the severing of actin filaments by the action of cofulin, which occurs predominantly during the unstretch phase. So as you go through the phases of a response to some sort of perturbation, the cytoskeleton behaves differently. It changes its material phase, and other things happen as well. Number 6, cells comprising a confluent epithelial layer tend to migrate collectively along orientations that minimize local in-plane intercellular shear stress, or what they call plithotaxis, and tend to exert tractions pulling towards any nearby cell-free void, or kenotaxis. So basically, as cells, epithelial cells migrate collectively along different orientations, they try to minimize, plithotaxis, and tend to exert tractions that lead to this kenotaxis. So they're basically minimizing shear stress and pulling themselves towards any sort of cell-free void. So they're pulling themselves towards open space. They're minimizing shear stresses, and so they're sort of amplifying their migration. Number 7, the confluent epithelial layer, when mature and unperturbed, tends towards a solid-like collective cellular phase that is jammed. In such a jammed phase, cells are crowded and trapped by their neighbors, and thereby become immobilized and stuck in place. So the jamming phase is where you get this critical level of density of particles, or in this case cells, epithelial cells. They become jammed even when trying to migrate, and so they become immobilized and stuck in place in the sheet. But by a variety of means, such a layer can undergo an unjamming transition, or UJT, wherein the cellular collective fluidizes and migrates much as a flowing fluid. So you get these transitions between this jamming where they become stuck, and then this unjamming where they become unstuck. And so this is managed by a single parameter that we can measure, and but any fluid, any sort of migratory process of cells undergoes multiple jamming and unjamming transitions as the sort of the environmental conditions require. In doing so, cell shapes become systematically more elongated and more variable. Moreover, cells with the unjammed layer retain a purely epithelial phenotype, including intact cell-cell junctions, intact barrier function, and no evidence of mesenchymal markers. So the UJT therein is distinct from this epithelial to mesenchymal transition. So these phase transitions, these jamming phase transitions, are independent of transitions between epithelial and mesenchymal cells. So this is interesting because we can decouple this process and understand the mechanisms. And so, they retain a purely epithelial phenotype. So we can look at the phenotype and say that there's no difference in the phenotype between jamming and unjammed. And so this is interesting, especially with respect to some of the stuff we've been talking about, again, with biotensegrity, that, we have to consider maybe this is cell type independent, or maybe there are some really interesting properties of cells that we haven't really discovered. So, and then finally, number 8, the confluent epithelial layer ordinarily stands poised in a disordered state that is just at the brink of unjamming, ready to unjam, and migrate in response to cues associated with wound healing, embryonic development, tissue remodeling, or cancer progression. Proximity to jamming can be quantified by cell shape and shape variability. So this is where we talk about cell shape and being poised at the edge of a phase transition, where you like there's this bistability where the cell has a certain shape and it's ready to go, or has another type of shape and it's not ready to go. In this case, what they're saying is a confluent epithelial layer is always poised in a disordered state. So it's sort of at the edge of chaos, operating at the edge of chaos. And it's just at the brink of unjamming. So if it's sitting in this confluent layer, stuck in place, it's ready to unjam when it's needed. And so this is key to this sort of plastic response. It maintains integrity when it needs to be, but then if there's a plastic response that's needed, the cells can be recruited and they can change their shape and undergo this phase transition. So this is really interesting work, and I think these points get they talk about them more in the paper, and I don't want to get very deeply into this, but I think they're very interesting for follow-up, especially with respect to biotensegrity and related research. So thank you for paying attention, and I hope you learned something.